In this short talk, I'd like to describe some work we've done recently on using chemically powered motors to activate filament network systems. This is work done in collaboration with Lian Xiao from Hangzhou. So I'm going to begin with a brief description of the synthetic motors that we're going to be using in these applications. And in addition, I want to point out some of the close connections there are in the theoretical underpinnings of both the chemically powered synthetic motors and molecular machines that inspired the application that I'm going to be talking about today. In the last part of the talk, I'll discuss active functional materials that are actually activated by molecular motors and the mechanisms that we use to control the dynamics of these networks. So I'll begin with a few words about diffusio-phoretically powered motors. These are the ones that I'm going to be using in these applications. This is an old topic now, but I want to point out some of the features that I'll be using in this application. So just a brief word about the mechanism that these motors use to move. It's a diffusiophoresis. And in these motors, there's a catalytic reaction that occurs on a portion of the motor surface. So on the right, you see a Janus colloidal motor. This is from Samuel Sanchez's work. It's a silica sphere half coated with urease, the enzyme urease. And that enzyme converts urea to carbon dioxide and ammonium hydroxide. I want to point out that generally these chemical reactions occur reversibly. And this reversibility, of course, is a consequence of the underlying microscopic reversibility of the dynamics of the system. Now, in these systems, generally the fuel and the product that's produced in the reaction interact with the motor through different intermolecular potentials. So as a result of the asymmetric concentration gradient that's generated on the surface of the motor and the difference in the intermolecular potentials, this gives rise to a body force on the motor. But since momentum in the entire system is conserved, the momentum balance leads to a fluid flow in the surroundings, and that fluid flow basically gives rise then to a force on the motor, a diffusiophoretic force that I've called F sub SD. And in the lower left-hand corner, you see an example of the streamlines of the fluid flow field that are generated by such a catalytic reaction occurring on a Janus motor. You can see it's quite complicated, particularly the near field flow can be quite complicated and depends sensitively on the geometry of the motor and the nature of the chemical reaction that occurs there. I'm also going to be talking about another type of Janus motor, a sphere dimer motor, where the catalytic portion is now a sphere and the non-catalytic portion is an attached sphere. But the physical underpinnings are the same for both of these motors. So I mentioned that the motor reactions, in general reactions, as a result of the underlying microscopic reversibility of the equations of motion are reversible. And so directed motion is really only possible if the system is out of equilibrium. There's no net motion in equilibrium. So I want to consider some of the implications of this. So the chemical reaction A to B that's occurring on the motor occurs with forward reaction rates K plus and reverse reaction rates K minus. And the standard mass action rate law for the net production of product in the system, little n, so W is the reaction rate, is just the mass action law form. It's the forward rate coefficient times the concentration of A minus the backward rate coefficient times the concentration of B. And the kappa plus and kappa minus are the rate coefficients per unit surface area. Now, to drive the system out of equilibrium, you have to couple it to reservoirs that actually alter the concentrations away from their equilibrium values. So I call those CA and CB bar. And so these are reservoirs with fixed concentrations of these species. You can also have a non-equilibrium state generated by out of equilibrium reactions in the bulk of the fluid. And some of the simulations I'll show you are done that way. But this is the general idea. When you work out the expression for the diffusiophoretic force, you can show that it's directed along the axis of the motor, that's U, and it contains a prefactor that depends upon the intermolecular potentials and characteristics of the reaction diffusion equation. But the part I want to emphasize here is that it also contains a factor that looks like the usual mass action form, but with the reservoir concentrations. And this term is actually proportional to the chemical affinity of the reaction. The chemical affinity is just the difference in the rate coefficients, and here I've divided by KT. So this is the term that's responsible for driving the system out of equilibrium. Because these concentrations are not the equilibrium concentrations, I get a non-zero diffusiophoretic force. It's also possible to subject these motors to an external force, in which case there's a second affinity that comes into play, which is the external force divided by KT. So because of the underlying microscopic reversibility and the fact that the system is driven out of equilibrium, as I've described, it turns out that the underlying description of both molecular machines and these diffusiophoretically powered synthetic motors has a very similar structure if you look at it from a fluctuating thermodynamic point of view. Just in general terms, both molecular machines and synthetic motors, they convert chemical energy into regular motion, they operate out of equilibrium, and they work in spite of being subjected to very strong fluctuations. The crucial part that I want to focus on is the fact that the reactions are reversible, as I said, and this gives rise, in the presence of external forces, to a coupling between the chemical synthesis and the external forces or torques. This is very familiar in biology. For instance, in the ATP synthase motor, it consumes ATP and causes a part of the motor to rotate. This is what drives flagella, for instance. But in additionally, you can actually apply an external torque, which in biology is done by a proton gradient in the system. And that causes the motor, instead of consuming ATP, it actually makes ATP from ADP. And so this is how the cell manufactures ATP. So this coupling between synthesis and external forces of torque is very common. And although I haven't described it in detail, exactly the same phenomenon comes true in these synthetic powered motors. But for today's talk, what I want to focus on especially is one feature of the expression for the diffusiophoretic force. And that is that its sign depends on the forward and reverse terms in this expression involving the reservoir concentrations. So K plus Ca bar is larger than K minus Cb bar. I have a positive affinity. That means actually that the motor moves in a forward direction. But if the reverse is true, if the affinity is less than 0, then the motor moves in a backward direction. So simply by changing the reservoir concentrations, or the concentrations of the feed species in the system, I can make the motor move forward or backward. Very similar to this molecular machine rotating clockwise or counterclockwise. So now coming to networks, in the biological realm, the filament networks in the cell are crucial, and they are responsible for a lot of the mechanical functions in the cell. And they're active networks because they're activated by molecular machines that move along the networks and change their dynamics. So in this case, I have a picture of an actin filament network, and there are myosin motors that activate it. So recently we've been interested in whether synthetic motors can operate in filament networks in a similar way. And this is a study recently that we did in PR Research about chemically powered motors that move on filament networks and change the dynamics of the network. But in today's talk, I want to instead focus on active functional materials. And let me start with an experimental example from Yoshida's group, but also a lot of work has been done by Balazs's group and others. It's an active area of research. So these are hydrogels that oscillate. So in this case, you have a hydrogel with ruthenium catalysts attached to the filaments. These are the red dots. And the surrounding medium contains the Borisov-Zavochinsky reaction, which is a reaction that typically runs out of equilibrium and shows a lot of interesting nonlinear dynamics, including oscillations. And the catalyst for one of the steps in the reaction is ruthenium. And so as this reaction in the bulk oscillates, it actually changes the hydration properties of the gel, and the gel goes from an expanded to a collapsed form. And these are just different ways of building the gel. And so what you see, if you look at the characteristic lengths of the gel, you see that as the reaction oscillates, the gel goes from expanded to collapsed forms. And these gels are actually thought of as potential applications in robotics and drug delivery, where drugs can be trapped in the collapsed form and released in the expanded form. So now coming to the question of synthetic motors, I want to consider the case where you have filaments which are activated by embedding these small dimer motors, diffusio-phoretically propelled, as constituents of the motor. In this particular case, I have a 3 pointing to the right, the catalytic spheres point to the right, and on the other half, the 3 motors have their catalytic spheres pointing to the left. I should say there's been a lot of work on similar filaments. A lot of it's been done in India, and some of the references are below, including work by Sneha Thakur, who's our one of the organizers for this symposium. In any event, the interesting point I want to mention is that not only do these motors serve as the activating elements in the filament, but by controlling the reservoir concentrations, I can make the motors move either forward or backward. If the motors move forward, you can see that they tend to actually crumple the filament, whereas if they move backward, they tend to expand the filament. So just by controlling the reservoir concentrations, I can induce a mechanochemical coupling in the filaments that causes them to change their conformations. So this can be used to actually design filament networks now. So now I have a large collection of active filaments, and if the external constraints are such that I have forward-moving motors, then the filaments tend to form a compact network, and this shows the average end-to-end length in the filament network of a filament, and you can see the length decreases if I start from the expanded form. You can also see there's a strong clustering, which is related to the clustering properties of active motors. On the other hand, if the motors move backward just by tuning the environment, then I have an expanded network with no clustering. And the upper curve shows the, again, the mean end-to-end length in the system. And this middle curve is actually an inactive network. So now you can actually control the dynamics of the network by controlling the constraints on the system. And in this example, I'm actually controlling the constraints by using bulk phase reactions that are out of equilibrium, as indicated in this diagram here. And so if I make these reaction rates oscillate, I oscillate the constraints. And this is a plot of again, the average end-to-end length in the network, and you can see the oscillatory change in the network undergoing expansion and contraction. So these pictures are very reminiscent of what you saw for the active gels. So I just wanted to point out a few of these features. There are a lot of possible extensions of this work and applications. The main take-home messages are that basically, once you understand the underlying statistical physics of these motors, you can use this information to actually design active networks and consider different ways to control their properties. So my feeling is that these active matter systems will continue to be studied in the future and hold a great deal of promise.